All right, moving on to chapter 8. Chapter 8 seems long, but I'm going to cut it down a lot. Okay, so don't freak out when you see all the slides and stuff. Okay. So where is chapter? Okay. Oops. There it is. Okay. So, slideshow from the beginning. Okay. So chromosomes. Okay. So the big picture. Okay. So the central dogma of molecular biology. DNA goes to mRNA, goes to protein, which has a function. Okay. When you mutate the DNA, the reason why mutated DNA is so dangerous, okay, DNA is where the information actually is. So you mutate the DNA, you change the mRNA. You change the mRNA, you change the protein. You change the protein, you could potentially change the function. Okay. All right. So, big picture, page 2. So, if you make base substitution mutations, you change a single base. Okay. You make a frame shift mutation, you read the whole thing wrong. Okay. Much, much, much worse. Okay. Okay. And the operons, which we'll get to in a little bit. Okay. Okay. Alteration of bacterial genes and gene expression. Okay. This is important. This is what I want to focus the whole lecture on. Changing the bacterial genes can cause disease. Okay. So we can change it and prevent the disease. Okay. Or even better, we can take what our knowledge of bacterial genetics, screw around the microbe and make them do what we want. And we do that for not only medicine, we do that in different industries, the oil industry, et cetera, even the freaking nylon industry is pretty cool. I'll get into that a little bit as well. Okay. Okay. So definitions. Okay. Make sure you understand these definitions: genetic, genome, chromosome, gene, genetic code, genotype, phenotype, genomics. Okay. All of these are definitions. Make sure you understand this slide. Okay. Genetic code. Okay. Okay. Phenotype and genotype. Make sure you know that too. Okay. Okay. This is a really cool picture. I really like this picture. Okay. The string that you see around that bacteria is the DNA of that one bacteria. Okay. If you unravel that thing, it is one long circle. Okay. That it's only been like folded inside the bacteria. Like you compress it all together, it actually folds into that bacteria. Okay. That's why it's such a cool picture. Okay. Okay. The flow of genetic information works 2 ways. One is vertical. Okay. Vertical is father to son or mother to daughter. Okay. The flow of genetic information from one generation to the next. Horizontal genetic transformation is movement of DNA from 2 living organisms to each other. Okay, so in the same generation. Okay. Okay, the DNA replication we're completely skipping. Okay. The RNA and protein synthesis, we're skipping as well. Okay. We went over this in physiology, so hopefully you guys understand that from physiology. Okay. All right. Now, this we're not skipping. Okay. The regulation of bacterial gene expression. Okay. So make sure you know what an Operon is. Okay. So, okay. There are two types of genes in bacteria. Okay. Constitutive genes are expressed at a fixed rate. Okay, so no matter what the bacteria is doing at that moment, it will always make these proteins. Okay, other genes are only expressed as needed. So you have genes that are inducible, okay, and genes that are repressible. Catabolite repression, and we'll talk a little bit about, but not very much. Okay, so inducible genes are genes that are always off unless you want to turn them on. So when you induce, you turn it on. Okay, repressible genes are always on unless you turn them off. That make sure you understand the difference between those two, okay? Okay. Okay, repression inhibits gene expression, decreases enzyme synthesis or protein synthesis. Okay, it's mediated by repressors. Repressors are proteins that repress. Okay, I know it sounds obvious, but sometimes you have to say it. Okay, the default position of a repressible gene is on, right? So a repressible gene is always on unless you purposely turn it off. Okay, inducers are off unless you purposely turn it on. Okay. So the Operon model of gene expression, okay, in bacteria. Okay, first you have an Operon. Okay, when bacteria want to make genes, they usually don't want to make it one at a time. They want to make a whole bunch of genes at a time. For example, we'll talk about the lac Operon in a second. The lactose is a sugar that we eat. And this is E. coli. They study this in E. coli. E. coli do not often see lactose, but when they see it, they want to be able to use it. Okay? So there's 3 different genes that are necessary for them to actually process lactose. So if you want to turn them on, you want to turn them on all at once. You want to turn them off, you want to turn them off all at once. So you have an operator that's in charge of multiple genes at the same time. Okay, you have these 3 genes, Z, Y, and A. The name of the genes doesn't matter. Okay, now I have a promoter that will cause gene expression. Okay, and then I have an operator. Okay. Okay. Operator is a segment of DNA that controls transcription of structural genes. Okay, so here is my operator. Okay, like I said, for lactose, lactose is a sugar that bacteria will eat, but only if it's actually there, right? I don't want to spend time and effort making a whole bunch of enzymes to digest lactose unless there was lactose. Okay, otherwise I would just waste protein, and wasting protein is not a big thing to it's not a good thing for bacteria. Okay. Okay. So what do I do? Well, okay, there's a promoter that causes transcription. The promoter causes transcription. So what do I have to do? I have to physically block the promoter. Okay? So the RNA polymerase, the only thing you have to know about RNA polymerase is that if you let it go, it will make the proteins. RNA polymerase, it will make the RNA. Okay? Okay, so what do I do? Well, I transcribe a little thing, okay, called the repressor, okay? The repressor will bind to the operator, okay? And it will physically get in the way of the RNA polymerase. So the RNA polymerase physically can't get anywhere. If it can't get anywhere, it can't transcribe the gene C, Y, and A. Okay? So if there's no lactose, there's always going to be an active repressor. The active repressor will bind, okay, to the operator, okay? Okay, the repressor will bind, okay, the active repressor will bind to this thing right here. And now it literally gets in the way, right? So imagine like polymerase as a train. If you block its path, it can't continue and it just stops right there. Okay. Now, what do I do if I do have lactose? Well, if I do have lactose, that means I have lactose in the bloodstream. Or not in the bloodstream, in the bacteria cytosol. Okay? If I have lactose, so lactose is this little green blob right here. Okay? This green ball will literally sit, if I have lactose, I will literally attach to the repressor. It physically binds to the repressor. Okay? Now the repressor no longer works. If the repressor no longer works, then the repressor is no longer capable of binding to the operator. If that's true, then this has no problem going. The RNA polymerase has no problem transcribing the genes. Okay, I know it's hard. You probably want to listen to it at least twice. Okay, but if there is no active repressor sitting on here, there's no physical thing that blocks the RNA polymerase from working. Okay, so that is an inducible operon, also known as the lac operon. Okay, lac for lactose, because this was the mechanism that Jacobin would know to actually figured out on lactose. It works for a bunch of other genes as well, but it's primarily known for lactose. Okay. The Operon model of gene expression in reducible operons. Okay. Structural genes are always transcribed unless they're turned off. Okay. Let me go back a slide. Okay. Inducible operons, the structural genes are not transcribed unless the inducer is present. So make sure you know the difference between the two. Okay. Okay. So there is a amino acid called tryptophan. Okay. Tryptophan is an amino acid that the cell needs. Okay. So the bacteria can make it easily. Okay. But bacteria don't want to waste energy making things that they already have. Okay. So if there is tryptophan, I want to stop making new tryptophan. If there is no tryptophan, then I want to make tryptophan. Okay, I only want to bother. I only want to stop making it if there's enough of it. Okay. So very same thing. I have a regulatory gene, I have a promoter, I have an operator, and then I have the structural genes. Okay, A through E. Okay, again, the genes don't really mean anything. Okay. Now, the default for a repressible operon is that you can turn it off. The default is on. So the default is on. So I make a repressor RNA. The repressor RNA doesn't actually do anything because it doesn't fit in that little hole. And if it doesn't fit in that little hole, then RNA polymerase has no problem going through. Okay. Now, if I do have enough tryptophan, then tryptophan will bind to the repressor. Now the repressor fits inside the hole. And once the repressor fits inside the hole, then the polymerase can't go anywhere. Okay? So it works the exact opposite way. Repressor operons are always on unless they're turned off. Okay? But hold on. I think I forgot to enable the computer sound. Share computer sound. Okay. Oh, I hate you guys. Share screen, share computer sound. Close down my presentation. That's it. Evil bastard. Okay, hold on 1 second.
Many bacterial genes are regulated in groups called operons. An operon consists of a promoter, a regulatory element called an operator, and multiple structural genes encoding proteins that are often involved in the same biochemical function, such as the catabolism of a specific sugar or the biosynthesis of an amino acid. All of the structural genes of an operon are transcribed into a single mRNA molecule, which is then translated into this entire set of proteins. Inducible operons are not transcribed unless they're activated by an inducer, while repressible operons are always transcribed unless they're deactivated by a repressor. The lac operon of E. coli includes a promoter, an operator, and 3 structural genes encoding proteins involved in the catabolism of lactose. The lac operon is an inducible operon, meaning that the genes are usually not transcribed. When lactose is available to the cell, the operon is induced or stimulated to produce the proteins of lactose catabolism. The E. coli chromosome contains a regulatory gene near the lac operon. This gene is constantly transcribed and translated to produce repressor protein. The repressor protein binds to the operator DNA at the lac operator, physically preventing RNA polymerase from moving beyond the promoter. When a repressor protein is bound, the genes for lactose catabolism cannot be transcribed into mRNA. When lactose is present in the cell, some lactose molecules are converted to allolactose. Allolactose binds to and inactivates the repressor proteins, preventing them from binding to the operator. The structural genes are transcribed, resulting in the production of the enzymes needed to catabolize lactose. Since allolactose induces or stimulates transcription of the lac operon, it is called an inducer. Notice that the inducer acts by turning off the repressor, not by actively stimulating the genes to be transcribed. When lactose is not available, the repressor proteins are active. The structural genes of the lac operon are not transcribed. This control prevents the cell from producing the proteins for lactose catabolism when lactose is not available.
Operon, this is the last one.
The TRP operon of E. coli includes a promoter, an operator, and 5 structural genes encoding proteins involved in the biosynthesis of tryptophan. The TRP operon is a repressible operon, meaning that the genes are usually transcribed. When excess tryptophan is available to the cell, the operon is repressed, ending the production of the proteins of tryptophan biosynthesis. The E. coli chromosome contains a regulatory gene near the TRP operon. This gene is constantly transcribed and translated to produce repressor proteins. However, these repressor proteins are inactive unless excess tryptophan is available. Once activated by tryptophan, the repressor proteins can bind to the TRP operator, preventing RNA polymerase from moving beyond the promoter. When a repressor protein is bound, the genes for tryptophan biosynthesis cannot be transcribed into mRNA. When more tryptophan is available to the cell than the cell can use, some tryptophan molecules bind to and activate the repressor proteins. The repressor proteins bind to the operator, preventing the transcription of the structural genes that encode the proteins of tryptophan biosynthesis. This regulation prevents the cell from producing excess tryptophan when tryptophan is already available. When excess tryptophan is not available, the repressor proteins are inactive. The structural genes of the TRP operon are transcribed and translated, producing the proteins necessary for tryptophan biosynthesis.
Okay. All right, so make sure you can answer these questions. Okay. Who knows where they might show up? Okay. Okay. Well, so catabolic repression inhibits all cells from using carbon sources other than glucose. E. coli loves glucose. Okay. If you have glucose, it won't bother doing anything else other than eating glucose. Okay. You only want to feed on glucose. It's like candy for children. Okay. 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 So if you just have glucose, it will grow better than glucose. Okay, because I don't have to. If I have enough energy off of the glucose, then I don't have to make protein for to digest all the other crap. I already have all my proteins for glucose, so I can use all the other proteins to grow. Okay, so it prefers glucose. Okay. Okay. Yep, all that micro switch stuff. Okay. Okay, changing the mutant. So one of the things you guys are probably, thinking about, worrying about, and it'll probably be in the news, is how fast viruses change, especially COVID-19. Okay, so that's mutation, right? Okay, so a mutation is a permanent change in the basic sequence of DNA. Okay, most mutations are neutral, some are beneficial, and some are harmful. Okay, so a mutation is neutral if there's no effect on the organism. It's beneficial if it has a positive effect on the organism, not on you. Okay, so COVID-19 developing a mutation that ends up killing a bunch more people is a beneficial mutation from the point of COVID-19. Okay, COVID-19 developed a mutation that no longer kills people and no longer transmits COVID-19 is a harmful mutation for COVID-19. Okay. So you always look at mutations from the point of view of the organism, not you. Okay, this is particularly important in microbial genetics. Okay. Okay. Base substitutions. Okay. Base substitutions are one single base substitution. You change an A to a T, etc., etc. Okay. Okay. A missense mutation is a base mutation that results in a change in a single amino acid. Okay. So here you have a glycine. Whoops. Here you have a glycine here changing to serine there. Okay. Usually those are not really harmful or they're usually really minor things and don't do very much. Okay. Missense mutations result in a stop codon being inserted much earlier. Those are much more problematic. Okay. A frame shift mutation is deleting one or two of the nucleotides. So I changed the entire reading frame. If you guys remember from physiology and stuff, we read the DNA code in 3-letter chunks called codons. If I delete one letter, then every character after it will be wrong. You change the reading frame. Okay, so after the lysine, after the lyse, every single amino acid will change. Okay. Okay, now chemical mutagens. Okay, so good thing about chemical mutagen, a chemical mutagen is what happens when you add a chemical and it screws around with either DNA synthesis or some other thing that introduces incorrect damage nucleotides to DNA. Okay, it usually causes bad things to happen to the bacteria or to the virus. Sometimes, however, that actually leads to good things. Highly unlikely, but that's happened before. Okay.
As living organisms reproduce and grow, they're constantly replicating DNA with a high degree of precision. However, inevitably, about once in every billion nucleotides that are replicated, an error occurs in the replication process, changing the sequence of nucleotides. Chemicals called mutagens can increase the chance of such mistakes. There are two major categories of mutagens: radiation mutagens and chemical mutagens. Different types of radiation have different effects on the DNA. Ionizing radiation, such as X-rays and gamma rays, can cause some of the molecules within cells to lose electrons, becoming highly reactive ions and free radicals. Some of these reactive ions and free radicals can combine with bases on DNA, resulting in errors in DNA replication, and therefore mutations. Even more seriously, these groups can react with the sugar phosphate backbone of DNA, causing breaks in chromosomes. Non-ionizing radiation in the form of UV light is also mutagenic because it can cause adjacent thymine bases to covalently bind to one another, producing thymine dimers. Such dimers can cause serious harm or death to a cell if they're not repaired, since these dimers prevent the cell from properly transcribing or replicating such DNA. Chemical mutagens have many different methods of causing mutations. Let's look at how 3 different types of chemical mutagens work. Nucleoside analogs are compounds that are structurally similar to normal nitrogenous bases, but with different base pairing properties. These compounds can become incorporated into growing DNA during replication, replacing their related base. Once incorporated, the nucleoside analog can inhibit further replication or cause mismatching in a future round of replication. For example, 5-bromouracil is a nucleoside analog of thymine, but it often pairs with guanine rather than adenine. Incorporation of 5-bromouracil can therefore lead to a base substitution mutation of a guanine for an adenine. Some chemical mutagens directly alter the structures of the nitrogenous bases of DNA. For example, nitrous acid can chemically alter adenine bases so that they pair with cytosine rather than thymine. During replication, this change causes a base substitution mutation in the daughter DNA. Some chemical mutagens cause small insertions or deletions of nucleotide base pairs, which can lead to frameshift mutations. Examples of such frameshift mutagens include acridine, a dye commonly used as a mutagen in genetic research, benzopyrene, which is found in smoke and soot, and ethidium bromide, which is used in laboratories to stain DNA. These chemicals are the right size and have the right chemical properties to slip between the stacked base pairs of the DNA double helix, producing a bulge in the molecule. During DNA replication, this bulge can lead to the insertion or deletion of one or more base pairs in the newly synthesized DNA.
All right. So, okay. So, okay. You don't have to know types of mutations and stuff. So, okay. You do have to know how we identify mutants. Okay. We can positively select them or we can negatively select them. Okay. Usually we do negative selection because it's easier. Okay. Okay. So you mutate a bunch of stuff. You take a bunch of bacteria. Okay. And then you grow them. And you grow them in a medium containing histidine. Okay. So they'll all grow. So they all grow and they all form little colonies. Okay. So now I'm going to take this velvet and we'll crush it on here, pick it up, and we'll stamp it on both of these. This will be the same thing as this. So whatever grows here will grow there. Okay, just in case it doesn't like pick it up. You can see it here. It doesn't pick up. Okay, but now here, this is a medium lacking histidine. If there is a missing spot here, it means that mutant has a mutation in the formation of histidine. Okay, so it can't make its own. Okay. Okay. The Ames test. Okay. Named after one of my old professors, Bruce Ames. Okay. Exposes mutant bacteria to mutagenic substances and to measure the rate of the reversal mutation. So I have a mutation that these bacteria, okay, cannot grow. Okay, so I incubate a plate. Okay, so here's the control. So I add liver extract to it, I grow it. Very few bacteria will be able to grow on the control plate. Okay, very few bacteria can revert to being able to process histidine. It's usually even less. It's usually like either none or like one maybe, and it'll look kind of puny. Okay, but if you have a mutagen in that tube, that mutagen can wreak havoc with the DNA. And when you wreak havoc with the DNA, sometimes you screw things up, like 2 wrongs make a right. And it makes that particular bacteria able to grow on histidine minus media again. And therefore, it'll grow. And therefore, you know that whatever you added, if it has a bigger cell count than the control plate, that is a potential mutagen. Okay? So it's called a reverse mutation test because you're reversing the mutation, so now it grows again. Okay? So make sure you understand what the principle behind AIM test is, okay? Okay, so gene transfer. So I want you guys to know what plasmids are. We'll skip transbosons, okay? So, okay, we'll skip genetic recombination. That's only useful in transbosons, so we're skipping transbosons, okay? Okay, vertical gene transfer. Transfer of gene from organism to offspring. Horizontal gene transfer is the transfer of genes between 2 cells of the same generation. So animals don't do this at all. So like an example of horizontal gene transfer is if I gave my gene to my brother, that doesn't actually happen in humans. It does in bacteria, and that plays a big role in antibiotic resistance. Okay.
The transfer of genetic information between organisms in the same generation is called horizontal gene transfer. 3 types of horizontal gene transfer occur in bacteria: transformation, transduction, and conjugation.
Make sure you know
all 3 mechanisms involve a donor cell that transfers a portion of its DNA to a recipient cell, which may incorporate the new DNA into its own, becoming a recombinant organism.
Okay, we'll skip transbosons. Okay, a plasmid is a self-replicating loop of DNA. Okay, that is not in the bacterial chromosome. Okay, they often code for proteins that enhance the pathogenicity of the bacterium, which is why this is important. Okay, and this is a plasmid. Okay. Don't worry about different types of plasmids, I don't care. Okay, we'll skip this thing about insertions of plasmids. Okay. Transformation. Okay. Genes are transferred to bacterium as naked DNA, just free-floating DNA. Okay. So here's transformation.
Some bacteria are capable of taking up fragments of DNA from their surroundings and integrating the fragments into their own chromosomes by recombination. Cells capable of taking up DNA from their environment are said to be competent. Only a few genera of bacteria are naturally competent, such as the Streptococcus pneumoniae in this example, but other bacteria, including E. coli, can be made competent through simple laboratory treatments. The process of transformation was first observed by Frederick Griffith in 1928 while developing a vaccine for Streptococcus pneumoniae. Griffith observed 2 strains of the species, an R strain and an S strain. The unencapsulated cells of the rough or R strain of Streptococcus pneumoniae did not cause disease. When mice are injected with these cells, their immune system efficiently deals with this invader and the mice live. Cells of the smooth or S strain of Streptococcus pneumoniae produce a capsule that allows them to escape phagocytosis by the body's defensive white blood cells. When mice are injected with these cells, the mice die. Heat can be used to kill the smooth strain of Streptococcus pneumoniae. When mice are injected with these heat-killed cells, the mice live. Surprisingly, when mice are injected with a mixture of harmless heat-killed smooth strain cells and harmless live rough strain cells of Streptococcus pneumoniae, the mice die. Live smooth strain cells can be found in the blood of these dead mice. The harmless rough strain cells must have gained the genes to produce capsules like those of the virulent smooth strain cells.
All right. So basically what happens is, look, you have okay. You have a bacteria, okay? Bacteria usually don't pick up naked pieces of DNA, okay? It does when it's stressed. But when you do, you stress it out. You shock it, you put it on ice, you do something to it. You even run electricity through it, but you shock it somehow. And now it's trying to find out how to survive. And so if there's DNA floating around, it'll pick up that piece of DNA and hopefully it survives. Okay? And that process is called transformation. So next slide. Okay, so here's the Griffith experiment. Okay, the only way that this mouse dies is if the live bacteria picked up genes from the dead bacteria. Okay, you also know about mechanism of transformation. Next one is conjugation. Conjugation is extremely important because this is how antibiotic resistant genes are spread, where you guys are going to be working in hospitals and stuff. So let's see what that means. Okay. Video. Okay.
E. coli cells containing an F factor, also termed the fertility factor or F plasmid, are capable of forming proteinaceous rod-like structures called conjugation pili or sex pili. Cells containing an F factor are called F plus cells and serve as DNA donors during conjugation. Cells lacking the F plasmid are F minus cells and serve as DNA recipients during conjugation. At the start of conjugation, a conjugation pilus extends from an F plus cell to an F minus cell and pulls the cells together. The cells come together and stabilize, probably through fusion of their cell membranes. The transfer of DNA begins. A single-stranded copy of the F factor is transferred from the F plus cell to the F minus cell. Each cell synthesizes a complementary strand of the plasmid, resulting in a complete copy of the plasmid in each cell. After conjugation, both cells contain a copy of the F factor. Therefore, both cells are now F plus cells and can go on to initiate conjugation with other F minus cells.
Okay, does that make sense? That's why antibiotic resistance can spread. Because in addition to the plasmid will have the antibiotic resistance genes, okay? And those can spread from cell to cell to cell, and they do not have to be the same type of cell. They do not have the same species, okay?
In conjugation, DNA is transferred from one living bacterial cell to another. The cells must touch one another, and after the process, both cells have their own copy of the transferred DNA. Since this process is best characterized in E. coli cells are usually used as models for bacterial conjugation.
No about HFR. Don't care. Okay, transduction. The last one.
Cells use DNA to store information, while proteins are used to perform the activities necessary to keep the cell alive. In a process called transcription, the information stored in a DNA molecule is copied into RNA molecules, which can be used to synthesize specific proteins. Cells use three types of RNA: ribosomal RNA molecules, which are used to form ribosomes; transfer RNA molecules, which deliver amino acids to ribosomes; and messenger RNA molecules, which carry the information for making specific proteins from DNA to ribosomes. Cells.
Okay, so basically, transduction is when bacteriophages screw up. So bacteriophage is a virus that infects bacteria, and when it's killing the bacteria, it picks up the pieces of DNA that it made as part of the bacteriophage. But occasionally that screws up, and instead of picking up bacteriophage DNA, it picks up bacterial DNA, and the rest of the protein, before it picks up the DNA, the rest of the proteins just make a big hypodermic needle, basically. And so it'll pick up the bacterial DNA and inject it into the next cell it infects. And therefore, the next cell will pick up new bacterial DNA, okay? Okay, so don't worry about specialized induction, blah, blah, blah. Don't worry about that. Okay, so don't worry about F factors and stuff. Just understand the basic steps of the basic differences between transformation, transduction, and conjugation. Okay. Okay, so mutations and recombinations that create cell diversity. Diversity is a material for evolution. Okay, I'm gonna relate this to coronavirus because we're in the middle of a pandemic. Coronaviruses are a big group of viruses that are slowly mutating into different viruses, okay? SARS is a coronavirus, MERS is a coronavirus, COVID-19, obviously a coronavirus. These viruses have been mutating in other animals, so we don't have any particular immunity against them, okay? All right, hopefully that ends this really long talk, but I cut out a lot of stuff. So hopefully you guys listen and focus on the stuff I talked about. All right.